I'm going to talk about research that's been done for the last 3 years or so at the University of Cambridge. I myself am a geneticist working mainly with dogs, but in all sorts of problems. So I wouldn't compare myself with Gerhard as an expert on brachycephalic obstructive airway syndrome. But this was a problem that was brought to me by higher up. Better? This was a problem that was brought to me by the lady standing on the left as you look at it of this group, a surgeon called Jane Ladlow. I was aware of it already, of course, but she has been actually the driving force in getting me to work on it. And the rest of the team includes a geneticist who's quite new to the group, a postdoctoral worker, Lajos Kalmar, the gentleman in the group, and a couple of students, the one in the center holding what is actually an exaggerated picture of a bulldog with his mouth, or an exaggerated sculpture of a bulldog with his mouth open. Naiche Liu has been absolutely central to driving this work forward. She's a PhD student. And the other hands-on person, Eileen Traconis, has joined us in the last year and has been working on some of the measurements I will show you. So I will take you quickly through these first few slides. They are your revision. You've heard all of this before. So to start off with, one can hear the dogs making noises. They make different noises depending on which is the most effective structure within the airway, but all of them will have some of the effects from all areas of the airway. So that first one was soft palate. Here's the larynx. That high-pitched note is very characteristic of larynx collapse. And then lastly, this snoring or snorting noise is very characteristic of nasal obstruction. And you can see actually that this dog has reasonably open nostrils, but it's blocked at the second site, which Gerard mentioned. And again, this is revision. So the positions that are commonly blocked are in the nostrils, in the turbinate structures, in the nasopharynx, and in the soft palate, the swelling of the soft palate, and then where the arrow is showing here at the larynx. I'm not going to take you into detail because that's already been covered. So what I did want to talk about was there is a problem in this field, which is that if you take your dog to veterinarian A, they will listen to the dog and they'll say the amount of obstruction here or the amount of noise the dog is making is quite normal for the breed and you don't need to do anything. And then if you take them to another veterinarian, they'll say your dog has a horrible obstruction and may need surgery. And the position of where you decide to draw the line as to what is an acceptable amount of nasal noise or blockage, respiratory blockage, is different for different veterinarians. So I wanted to do genetics on this problem, and in order to do genetics, I needed to have a standard. I needed to have a way to decide whether the dog had obstruction or whether the dog, at least functionally, didn't have enough obstruction that we needed to worry about. And the way we decided to do this was direct measurement of the respiration. And what we essentially do is we put the dog into a box and let it breathe and measure pressure changes. And I should allow everybody to know, no, I'm not sure which of these is going to be the laser. Which one's the laser, Gert? Is it? Oh, yes, at the bottom here. Okay, I should let everybody know so they don't think we're suffocating these dogs, that there is an airflow in, and at the back somewhere there's an airflow out of the box. And we have carefully balanced fans to allow us the airflows to be exactly identical, and it's computer controlled, so we can still see pressure changes as the dog breathes. And what you get is a trace of the respiration, and this is quite a good one for a pug, and you can see that there's just a downward pressure on inspiration, upward pressure on expiration, and we can measure all sorts of little measurements within the respiratory trace. And so what we have done with that is started to try and develop a tool that will tell us when the dog can exercise normally and when it can't. And I'm not going to say too much about the exercise test because I know the next speaker is going to cover that because she's the lady who we've been working with in the French bulldog breed. But the test's non-invasive, so you don't have to sedate the dog. It's very accurate on the other hand. It produces objective data. We don't say this is a good trace or that is a bad trace. We've trained a computer and the computer says that. And it's calibrated, as I said, by looking at exercise performance of these dogs. So just very quickly, the exercise performance that we have is that we listen, and we have the two trained vets in the team listen to the dogs, listen for their breathing and chest noises before and after being trotted at 8 kilometers an hour. So this is a trot, not a walk, for 3 minutes, and that means it's about 400 meters. Why did we decide on that amount? It was purely because we wanted the dogs to get into respiratory deficit during the test, so we wanted them to trot and not walk. And 3 minutes was a convenient amount of time because a lot of these tests are done away from the university and in little village halls and so on, which have a football pitch outside them. So you can take the dog around the football pitch and you will do about 400 meters. Very simple. And so we then grade, and we've been grading into 4 grades, but I'm not going to go into these except to say that the good ones were simply differentiating on the respiratory noise and the inspiratory effort being made by the dog. And the bad ones, the BOAS plus ones here, were grading on changes in the noise and whether they refuse or whether they're unable to complete the complete exercise or unable to do it at the speed that we would like them to do it at. And so we regard this as a functional grading. If you're in these 2 grades, you may make a little bit of noise, particularly post-exercise, but you're able to perform in a way that is sufficient for you as a dog with an owner, unless your owner happens to want to run you at huge speed, which I think most people will not with a brakycephalic dog. And the sort of respiratory changes we see here are reasonably good examples of these 3 breeds. And what you can see is just that the bigger the dog, the bigger the traces, as you might expect, the bigger the pressure changes. But when you look at poor examples, initially you start to see perhaps a bit of tremor within the trace and a somewhat flatter trace with much more sort of spiky in and out breaths. And subsequently you can see things like complete collapse of the airway at some point in the cycle. And this can be at one end of the breath or the other, and this probably depends on whether this is at the nostrils or at the larynx. And you can also sometimes see very bad traces simply because they've got large spikes in them. And these seem to correlate with structures partially obstructing the airway and with turbulence being set up in the airway because of that. And so we have a variety of types, but the main thing is we have a computer to sort this out for us. And this is just showing how the computer is able to sort the BOAS minus traces. The little blue crosses over here are what we call grade 0. In other words, those dogs never make any noise at any stage other than normal panting during the run. They never make noise before or after the trace. The ones we think are acceptable are in yellow here. The lower grade of the ones which we nonetheless think aren't acceptable are in red, and the very bad ones at the top there in black. And so that shows us the computer is able to make assessments with very little disagreements from the vets, and these are vets who are very practiced at doing this. And we actually believe that probably the computer is better than the vet. That the respiratory trace at rest is telling you more consistently than the vet can do, who's been collecting these over a matter of a year or so. And so this just shows the accuracy of the test. So if you're up at the extreme top left-hand corner in these what are called rock curves, that means it's a very good test. It never gets it wrong. And if you're down anywhere else in the graph, then it gets it wrong from time to time. And this compares the exercise test data with the computer's data. And you can see for bulldogs, it virtually never gets it wrong. For French bulldogs, it's, again, it's very good. Slightly less good for pugs, but not bad. And it can do it with any brachycephalic breed. We put a different set of starting parameters in and say you're going to look at all brachycephalic dogs. For the breeds that we've concentrated on, it's pretty good, even if you don't tell it what breed it's looking at. So how are we using that? And this is the point. We need objective data about this. And what we're using it for is, first of all, to look at the prevalence of brachycephalic obstructive airway syndrome in our experimental groups. And I have to explain this is not a complete cross-section of the population at this stage. These are the dogs that it was easy for us to get at. And the dogs that it was easy for us to get at come essentially in three groups, two of which we've lumped together in here. The ones in blue at the bottom are hospital patients. So those dogs are generally severely affected with BOAS and are ones that have come in for advice as to whether to have surgery or not. And I've taken those out of the figures that I've shown up here. Then the ones in red consist either of a population of show dogs, so we go to dog shows to get these. We also invite breeders to volunteer. And so these are volunteer dogs, and we've had a great deal of assistance in collecting those from the breed clubs. And I've got to pay tribute at this point to what a great job Penny and colleagues in the other breed clubs have been doing to help us with this project. And then we have looked at, according to our criteria, the test of the dog going through the chamber. And these were the first 100 dogs in these first two breeds. And at the stage that I made this slide, we were at 66 bulldogs. We've now got a paper submitted which has 100 of each. What you see is that for the pugs, a large number of them, using that fairly stringent exercise test, we would consider as BOAS positive. 60% of these volunteer dogs, a little under half of the French bulldogs, and again, a little under half of the bulldogs in our hands, of these show dogs, were affected. We then looked at the age of onset, and again, this is a biased sample of the population in the same way as the last. We don't have access to all age groups. In particular, we don't have access to very many old dogs. But we've been able to look at, and now these are much bigger numbers. This is more or less up to date. We've been able to look at much bigger numbers, and we can see that under the age of 1, we've got something like 40–45% of pugs affected, and a much bigger group unaffected. But by the age of 3 or above, we have only about 30% of pugs unaffected. And for French bulldogs, again, you can see the same trend. With age, this gets to be a disease that's more measurable, and the same for the bulldogs. We then looked at the effect of weight. And what we've done here is use a standard body condition scoring scheme, which grades dogs between 1 and 9. And most of our dogs actually occur between 5 and 8, it has to be said. As I say, most of them are show dogs. And so you can't take a really, really fat dog to a show. And certainly with these breeds, judges seem to like the dog to have at least some fat, certainly in the UK. So I think probably the largest category of our dogs are graded as 6 by us. And what you can see here is that the French bulldogs, again with Penny leading the way, actually we had very few dogs which had scores above 7. Or 7 or above. They have got their dogs down now so that nearly all of the show dogs are 5 or 6, occasional 4s, occasional 7s, but nothing much else. And so we weren't able to get a great deal of significance in the result for them. But certainly for pugs, we're able to show, and this is again a large number of dogs and has a p-value with several noughts in front of the one. That's to say a very low probability of occurrence by chance. We were able to show that if you're fat, you're more likely to be affected with BOAS. And this is corrected for age. And similarly for the French bulldogs, in as far as we could find fat dogs in our groups, if you were fat, you were more likely to have the condition. Surprisingly for us, that has not been true for bulldogs. And we're not quite sure why that is, but we think it may be because in bulldogs, it appears to us that quite a lot of the owners who don't have the problem have been coming forward, and perhaps less of the owners who do have the problem have been coming forward. But that is purely hearsay. We haven't got any proof of that. Nostrils, we've heard about the effect of nostrils, and so I will proceed straight on to just showing you the data. First of all, we have dogs with stenotic nostrils, particularly in French bulldogs, quite a lot, more than half of the dogs are pugs, and slightly under half of the bulldogs, using criteria of external appearance. Here, the trend is very clear. If you've got stenotic nostrils, this is not a surprise to anybody, but it's very clear that if you do have poor nostrils, you're more likely to have BOAS. And this is particularly true in French bulldogs, where the odds ratio is actually more than 10, that you're more than 10 times as likely to have BOAS if you have stenotic nostrils than if you don't. Confirmation, we've started to do some work on. I'd say that this is not really complete yet. We just wanted to show, by showing the skull case and the cranial vault of the skull case compared with the whole skull, how very different in conformation these different dogs are. And therefore, you wouldn't necessarily expect the same solutions in terms of conformation to apply to each breed. And I think that's one of the take-home lessons that I would like to impart in this talk. So we have been using soft tape measures, just measuring externally the dogs. Generally it shows, and these are rather excited dogs very often. They're being handled by new people, there's lots of other dogs around, and therefore what I wanted to preface what I'm going to say by is that there are some difficulties with taking these measurements, and that therefore the accuracy isn't as good as it might be, and we're probably missing a few things because some of the variation we see is just variation because the dog's moving. And we also know there's some variation between people taking the measurements. But having said that, we have still been able to find some correlations. So this little chart here, p less than 0.01 in red, and then with 1 less 0 in pink, and between 0.05 and p equals 0.01. These are probabilities that what we're seeing is a real difference. This is a statistical check that scientists do on their work. And anything that has a p-value which is less than 0.05 is generally accepted by science as being unlikely to be a random effect. But I've also put in, because that isn't a magic number, it's just the way convention is, I've also put in events that would be perhaps a little bit more probable than that, but maybe showing us something. And this is because of the variation I see in the measurements. So taking this pug, we notice that the width, actually as measured between the eyes rather than the whole skull width, seem to be correlated with the presence of disease. The neck girth, this has been reported by others. We only found it as a trend in this breed, not as what would scientifically be seen as proven. And actually also the neck length, we find that if you have a shorter neck, that's bad news. So that's the pug, and here comes the French bulldog. And here we were able to find rather stronger correlations. French bulldogs are a little bit more variable in conformation in the UK than pugs are. And again, the neck girth, the skull width this time rather than just between the eyes. The craniofacial ratio, that's the ratio of muzzle length to length of the cranium, length of the top of the skull. And also actually the body size. If you're a big dog, you're slightly more likely to have BOAS if you're a French bulldog, at least in our population. So that's the French bulldog. Put that one away. And the bulldog, and I'm told this is a very bad bulldog, but I apologize for that. It was just a drawing I could find on the web. Cranial length seems to be important by itself. If you have a shorter cranium, you're slightly more likely to have problems. And also the neck girth is a problem, and the skull width. Now I should say that all of these I've adjusted for the body condition, for whether the dog is overweight, and I've adjusted for the gender of the dog. Okay, so putting that one away. We're also able to assess objectively whether surgery works. And this is actually surgery on the turbinates. In Cambridge, we do surgery on the other areas first. We regard the turbinate surgery as being more drastic. And we look at whether the dog has recovered using these objective grounds before doing the turbinate surgery. But this is a nice example because it isolates just one bit of the total surgeries that Gerard talked about. And I won't go into it very much, but here we've got the pre-surgery condition, and here we have the post-surgery condition, and this grey line is where the computer tells us these dogs will be able to run 400 meters at 8 kilometers an hour. And what you can see is that it does work for most dogs. So we now objectively can say this is a great piece of surgery, and we're very pleased that we were actually taught to do it by Gerard as well. So we're very pleased that he's helped us with this project. And lastly, I come to the genetic study. And again, we only started this in October, so this is fairly preliminary. Why do we bother with genetics? And you might say that the simplest way would be to breed dogs with long muzzles and to forget about all the breeds that you hold so dear and say, if a high proportion of these dogs have problems, we ought to just abandon them. I'm not going to take sides in that argument. I'm just going to say there is another way. And why not do that? Because you guys don't want that. You want to have distinctive dogs. And so that's very fair. And furthermore, even if we tried to compel you, there might be countries within the European Union that would be willing to do that. But they certainly don't include Britain, and they probably, I would think, although I don't live there, don't include the USA. I've worked in the USA for a number of years. I'm pretty sure that it's not going to happen. So they're not going to be legislated out of existence. And indeed, in Britain and in the States and in other countries in Europe, these breeds are increasing very rapidly in popularity. And Penny, I'm sure, will tell you, are you going to say any more about this? Yeah. Okay. So just about as fast as they can be bred, they're being registered, and there's no selection going on at the moment. They're very cute. I particularly like this little couple. And also a lot of celebrities have them, and there's an awful lot of advertising, at least in Britain, which relies on these dogs. And it's very hard to compete with that. We don't have millions of euros to throw at the problem. They're hard to select against directly, partly because you don't know at breeding age whether your dog eventually might have an airway problem, and partly because, as I've shown, there are a diversity of physical characteristics which correlate a little bit with the disease. But actually, none of them are very big variations, and you can have dogs with some of those variations and not others. So genetic testing might offer improvement without radical shape change. We know that there are dogs with fairly identical external characteristics that have BOAS and others that don't. It offers availability of a test before breeding the dog. And it has, at least in the UK, quite good acceptance in other conditions by breeders. Breeders do tend to believe and go along with genetic tests. Obviously, there are problems with genetic testing, including cost, and that's something we might talk about this afternoon. So how do we find genes that cause inherited disease? Well, let's imagine there's an ancestral chromosome in which exists. So a piece of DNA, and these rods represent there's a double helix with bases. Bases are the information-carrying bits of the molecule. And let's say it's got a base which we'll call W there, another called X there, another called Y there, another called Z there. And that's in one parent. And another parent has slightly different variants at those positions, which we'll call A, B, C, and D. So now a mutation comes along that causes a disease. Doesn't have to be BOAS, it can be anything. And then there is recombination between these two chromosomes over the generations. Both exist in the breed. You do breedings, and bits of these get recombined with bits of those. That's to say that there is genetic exchange goes on over generations. And you end up in a situation where perhaps the end bit of A is now joined to this chromosome that's taken the mutation, and the end bit of D is joined there. And so this mutant, which when it first happened was connected to W, X, Y, and Z, is now connected to X and Y, but in some dogs there'll be a D there, in some dogs there'll be an A there, in some dogs perhaps the original mutation has survived. What that gives you is something that looks like this. Along here, we've got all the variations within the genome, and we routinely now use 170,000 variations across the whole genome in order to look for areas which are linked to a mutation. And I'm sorry, I can see some of you are thinking this is very difficult, and I apologize that I have to talk in English. My only 2 visits to Sweden have been for the Swedish Kennel Club, so I have an excuse. I'm not like Gerard. Anyway, this shows some work I did, or my lab did a few years ago. And what you can see here is a stack of dots in a particular place. And each of these represents a different, what we call polymorphism, a difference between A and W, B and X, and so on. And those exist not because all of them cause the disease, but because when you compare diseased animals with non-diseased animals, they're all close to the place where the disease gene is. So this gives you a marker for the diseased gene. What it says is at the top, the 2 groups, the group with the disease and the group without the disease, are very different. At the bottom, they're essentially the same. So here you've got a nice single gene causing a peak, and somewhere under that peak is the mutation that causes the disease, which you then have to go back and search for across the genome, across that little bit of the genome. So here's the initial data on the French bulldog, and this data, I have to tell you, we got on Thursday of last week, 9 days ago. So it hasn't been very much analyzed yet, and it's only half of our French bulldogs, and we've now got all the DNA samples we need for the other half, and indeed also to do the bulldog and the pug in the same way. But we haven't had those results back yet from our genotyper. And what you can see is I've put in a green line and a red line, and these are different ways for this technique that different authors have used to look at the significance of the result. And the main thing is that the more stringent one is the green line, and if you see dots which are actually over peaks, I think, can't see the 21, that peak there extends up to there, for example. If you can see dots like that well above this green line, then you've probably got something to work with. So the stage we're at now is that we've got 3 and maybe 4 genes, if you accept this way of doing it, to work with. And so far, we've only got half our data, so we're confident that our sensitivity is going to go up. So that's really where the genetics is. I'll draw your attention just to these little red arrows, where the thing we're measuring dips, and it gets closer to the 0 line. That's very interesting. That says in those areas, all French bulldogs are the same. And the red arrows are actually the positions of genes that others have said are to do with skull shape. And that says there are major parameters of skull shape that we're not going to see varying. And any ones that we do see varying are actually novel if they're connected with skull shape. But they could be connected with other things. So I've mentioned skull shape, and I've said that other people have done work on skull shapes, and some loci are already known. There are more loci which are already known in humans. But we also know from our own data that you can have the brachycephalic shape, you can have those genes that have already been discovered, but not have BOAS. So what else is going on? We've talked about soft tissues, the soft palate and its development has been talked about. We've talked about obesity. Both of those are partly determined by environment, partly by genetics. Similarly, cartilage composition, some of the changes in cartilage may be secondary to the disease, but there may also be some primary changes. Also, there may be genes in the respiratory center. I'm thinking particularly of those that are implicated in central sleep apnea in humans. We don't know if that's going to apply here, but actually one of our little blue arrows makes me suspicious that it might. In fact, the way tissues respond to being damaged may also be important. So if you respond by losing your form, by producing a lot of what's called fibrosis, a lot of scar tissue, then individuals may differ in that as well. And lastly, and here there are genes known already, many of these dogs are very wrinkled. Wrinkling is to do with a number of things that are specific to skin, but also a number of things that occur in other soft tissues. And there are well, there's one major gene known in that. And I think, Oki, you're a co-author on that paper, aren't you? So Oki will tell you about that. How do we use the information? Well, first of all, we need to determine how often different gene combinations occur and how often they are found with BOAS. Once we've done that, we can then estimate what are known as genomic breeding values by taking just the DNA type from a dog when it's young, when it's a pup, and being able to predict whether or not, if that dog is kept well, what proportion of dogs like that will have BOAS. Now, that doesn't mean we'll be able to say yes or no for every dog. It means that we'll be able to say, your dog has a particular combination, that means it only has a 5% chance of getting BOAS. Your dog has another combination, that means it has a 50% chance of getting BOAS. That's the sort of test that we might be looking at. And so population geneticists can then use that to develop a system similar to the estimated breeding value system that's used in hip dysplasia by the UK Kennel Club, which will allow you to estimate whether this is something you need to allow for in your mating choices. It also means that if the animal is vulnerable, we can tell you that, and you can look after that animal particularly carefully in terms of temperature, exercise, and so on. So this is just a summary slide saying what I've said, which is that objective assignment of dogs to BOAS plus or BOAS minus allows us to look more deeply at conformation, at therapeutic outcomes, and at genotype. Breeds are different from each other. We haven't seen that yet genetically, but we do see it already in conformation measures. BOAS prevalence reaches the maximum only above 3 years old, so you've got to have your assessments done not just once when the dog is a year old, but perhaps after that if you're looking at respiratory data. If you're looking, I think, also at exercise testing or whatever other tests may be applied. No surprises, obesity contributes to BOAS, nostril stenosis contributes to BOAS, aspects of conformation influence BOAS. We are doing, currently, we're doing some work with CT scans to look at soft tissue development, but I haven't presented results from that today. A proportion of all breeds, including those with short muzzles, do remain unaffected by BOAS. We've done a number of chins where the muzzle actually recedes from the top of the cranium, a number of Japanese chins, which are unaffected by BOAS. So there is no lower limit to this. Some genetic causes of brakycefali have been identified but are not being used to eliminate BOAS. BOAS can occur in dogs with brakycephaly, but it doesn't have to. This was really just an exercise in finding out how the skeleton develops. And a genetic examination of BOAS, that is what Cambridge is doing, but it's too early yet for us to draw conclusions. So with that, which I hope has kept us more or less to time, I think it has, I should acknowledge that there are a number of funders who've contributed to this, particularly the UK's Kennel Club Charitable Trust, but also a number of funds from within Cambridge and outside it. And I should particularly acknowledge the contribution of breed clubs and dog owners. If we're going to move forward on this disease, you guys are absolutely essential to helping us do that. It cannot be done without cooperation. So we want to find ways in which we can cooperate with you. Maybe to move things slightly, it may involve slight conformational change. It may be that when we've sorted out all the genetics, there's not really very much of that and there's more going on internally, but we don't know that yet. I have to acknowledge a bunch of statisticians who helped us with all the different techniques that we're using statistically and with the computing. I've already mentioned who the team are. So with that, I shall stop.